Thank you so much for that introduction, and it's an honor to be here and to present my PhD work. A PhD student at the Rockefeller University with Gabrielle Victoria. And as already introduced, I'm going to talk about molecular fate mapping of serum antibodies in the context of original antigenic sin. Since the pandemic, I don't think I need to explain the importance of antibodies to anyone, and perhaps not even this graph, which describes the kinetics of antibody responses upon repeated exposure to the same or similar antigen. This is the reactivity in the serum as you are exposed multiple times. So the key of this graph is that after the first exposure, subsequent exposures are way more robust and rapid due to the presence of immunological memory that forms during the primary response. Taking a step back, antibodies are made by B cells, and each B cell encodes a unique antibody. And upon primary exposure to an antigen, be it via infection or vaccination, antigen-specific B cells will be activated, and a subset of them can improve their antibody for the antigen in a mini evolution process, which happens in so-called germinal centers, mostly in lymph nodes. So what happens in this germinal center reaction is that the antibodies affinity mature, and one of the main consequences is that affinity improved antibody secreting plasma cells are formed, as well as memory B cells. And it is these memory B cells that can quickly respond when the antigen is seen again. In a previous study, when I started my PhD, we used various fate mapping mice where we could label with fluorescent proteins the germinal center B cells and their memory progeny with different colors. As for instance illustrated here in this lymph node, where you can see these clusters of fluorescent cells, which are the B cells that are undergoing this mini evolution process. So then, having labeled the memory progeny, we simply asked, how do these respond upon boost? And as expected, of course, these memory B cells became very efficiently plasma cells. But at the same time, to our surprise, we found that boost-induced germinal centers, new germinal centers, were mostly composed not of memory from the first round, but rather a new repertoire of naive B cells. So there's the potential to induce new responses. So then after priming and boosting, you end up with a scenario where your memory B cell compartment consists of B cells originating from the primary and the secondary response. And then the natural question was, how do these different cohorts of B cells contribute to the serum effector compartment, which is what ultimately matters for most neutralization aspects. So in this context, original antigenic sin becomes important, because this is a phenomenon that was described in the 1950s by Thomas Francis Jr., which originally described in the context of repeated influenza exposures that there seemed to be a tendency for the immune system to respond best to variants of flu that were seen early on in childhood. So the best responses tended to be to strains that were the priming strain rather than the boosting strain. And there's been a large body of research ever since. But with our current day knowledge of the B cell response, we would then hypothesize that upon a repeated exposure to an antigen, the immune system is prone to reusing that first cohort of responder B cells, so the memory cells from the first round, at the expense of inducing a de novo response, a new response. And related concepts are antigenic imprinting and antigenic seniority. And to the way we define them, I'll try to explain that using these illustrations. So we know that memory is very efficient at becoming antibody secreting cells, and that is true in all of these models. Right? When you are boosted, in the red line, the memory B cells from the first round are efficiently boosted, because that is what memory does. But then the key question is really, to what extent do you also induce new antibodies that might be important for variants? And whereas imprinting and other theories pose that new responses are maybe less than the memory response, the key question for original antigenic sin is that new response really suppressed by the presence of memory? Is the memory so efficient that it makes the new response less optimal? So understanding the balance between these two is what we were interested in sorting out. But importantly, with existing technologies, distinguishing these two models is virtually impossible, because when you look at the reactivity in the serum, you're just measuring the total amount of reactivity, and you don't know what's old and what's new. So to overcome this, we developed a new molecular fate mapping tool in mice, where we essentially knock into the Ig kappa light chain of the antibody gene this construct. So we're knocking in a flag tag, followed by a stop codon, which is flanked by LOXP sites, followed by a downstream strep tag. And essentially what happens is that upon the expression of an enzyme called Cre recombinase, this locus is genetically recombined. such that antibodies go from being flag tagged to strep tagged. So this is how it would work. If you cross these mice to Cre that's specifically expressed by B cells in the germinal center, and that is dependent on the presence of a drug called tamoxifen, we can induce a response, have germinal centers that are flag tagged, and then give tamoxifen to recombine specifically at that moment in time all the B cells that are undergoing this process to become fate mapped with strep. And then, since it's a genetic change, the memory progeny will forever be strep tagged. And also when these memory cells from this specific cohort of B cells will be boosted and become antibody secreting cells later on, these antibodies that they're secreting will be secreting strep tagged antibodies. So using this system, we went straight to addressing our question of, do we see OAS, original antigenic sin? So the setup is to illustrate here. We're priming and then boosting twice, and we're labeling with tamoxifen with that first cohort of B cells. So what's happening is that we're getting a germinal center that's fate mapped. The memory B cells deriving from this response will be fate mapped, and also all the antibodies that will be generated in the future by this cohort of B cells. At the same time, when we're boosting, as I told you before, we know that the new germinal centers are mostly new cells, so they would not be fate mapped, they would be flag tagged in blue. And since we're not giving tamoxifen again, they will remain blue and will be reverse fate mapped with flag tag essentially, and also the future responses. So using this system, we can distinguish exactly the question: do we see the suppression of this de novo response, or do we see an accumulation of de novo responses originating from these new germinal center B cells? So then we started out immunizing with a simple model antigen against TMP KLH, and then we measure the antibody reactivity against this molecule. We see the expected pattern of boosting. And when we dissect this response with our fate mapping precision, remarkably, we find that in the red line, even after repeated boosting, most of the response comes from that original cohort of memory B cells, rather than from the memory that was generated in the second response. And you do see this new response coming up and down, but you can appreciate it's on a log scale. So where you quantify the percentage of the antibody reactivity, it's almost everything coming from that first cohort of B cells. When we use more relevant antigens such as influenza, or in this case SARS-CoV-2 induced by mRNA vaccination, we find essentially the same dominance of the primary cohort of B cells contributing to the serum compartment over and over again, akin to original antigenic sin as described. So this is all in the context of boosting with exactly the same antigen again and again. So what happens when you boost with variant antigens? And for instance, with Omicron, what's highlighted here are the mutations that arise in Omicron in the receptor binding domain of the spike protein, which is the main antigen of SARS-CoV-2. And when we boost the mice with this antigen, compared to the homologously boosted group, where we didn't see de novo response, in the heterologously boosted mice, you see a much bigger contribution of new antibodies to the response, originating from new germinal center reactions, resulting in a decrease in OAS. So here we have mice that have old and new antibodies, and then we can do other things with them rather than just measuring their reactivity to the antigen. We can also look at how good are they at neutralizing the virus, for instance. So we can fractionate the serum into old and new antibodies, and then we did, in collaboration with the BNASH lab, we performed in vitro neutralization assays, and we found that the new antibodies that are flag tagged were superior neutralizers, the drifted strain. On average, they were 7-fold better. And to understand why exactly this is, we collaborated with the Bloom lab, where we could pinpoint exactly what parts of the antigen are being targeted by the old and the new antibodies. So by the strep versus the flag. So look at the bottom are the de novo induced antibodies. And we found that these new antibodies that were induced specifically by Omicron were targeted new epitopes that were not covered by the strep antibodies on top. So they seem to be specifically targeting holes not covered by the existing antibodies. And thereby, the immune response seems to be really tailoring these de novo responses to new epitopes. To summarize, we made these molecular fate mapping mice that allow for precise quantitation of the contributions of different cell populations to the antibody pool. When we look at homologous boosting, we find a strong OAS suppression phenotype, indicating that it's the primary cohort of B cells that repeatedly are reused. When we boost with variant antigens, we find that this reliance on the primary cohort of B cells decreases as a function of antigenic distance, and that these de novo antibody responses are tailored for the variants. And overall, I think this study highlights the importance of the division between the effector compartment at the serum level and what's happening in the germinal centers, where the germinal centers are a platform for generating de novo responses. And those are only contributing to the effector compartment once the antigenic distance is great enough for them to be recruited. And with that, I would like to thank especially my mentor, Gabriel Victora, as well as people I collaborated with in the lab, especially Luca Messin and Marije, and the collaborators I mentioned along the way. And thank you so much for your attention.